Hello viewer, welcome to the YouTube channel FahadYounas. Today I will discuss the topic life cycle of bacteriophages. What are bacteriophages? Bacteriophages are viruses that infect bacteria. They have a complex life cycle that involves attachment, penetration, replication, assembly, and release. They can either lyse their host bacteria or integrate their genome into the bacterial genome, becoming a prophage. Stages of bacteriophage life cycle. Attachment. The bacteriophage attaches to the host cell. Penetration. The bacteriophage injects its DNA into the host cell. Replication. The bacteriophage DNA replicates inside the host cell and new phages are assembled. Attachment and penetration. Bacteriophages attach to specific receptors on the bacterial cell wall. The phage then injects its genetic material into the bacterium. The phage genetic material takes over the bacterium's machinery. Biosynthesis and maturation. Biosynthesis involves replication of the phage DNA and production of phage proteins. Maturation is the assembly of the phage particles and packaging of the DNA. The maturation process involves cleavage of precursor proteins. Release and lysis. During the release stage, new phages are assembled and matured. The mature phages then leave the host cell by causing lysis. Lysis is the breaking down of the host cell wall. Life cycle of bacteriophages. Bacteriophages, often referred to as phages, are various viruses that infect the replicate within bacterial cells. The life cycle of bacteriophages typically consists of two main stages, the lactic cycle and the lysogenetic cycle. These cycles determine how the phage interacts with its host bacterium. Attachment. In the first step of the phage life cycle, the bacteriophage attaches to the surface of the susceptible bacterial cell. This attachment is highly specific and involves interactions between viral protein, tail fibers or tail spikes, and receptors on the bacterial cell wall. Penetration. Once attached, the phage inject its genetic material into the bacterial cell. Some phages have a siren-like structure or tail and puncture the bacterial cell wall and injects the viral DNA, while others may use different mechanisms. Replication and transcription. Point A. Lactic cycle. In the lactic cycle, the injected phage DNA takes over the bacterial machinery. The bacterial cells down DNA replication and transcription processes and hijacked to replicate the phage DNA and produce viral components. Point B. Lysogenic cycle. In the lysogenic cycle, the phage DNA integrates into the bacterial chromosome, becoming a prophage. It can be replicated along with the bacterial DNA during cell division, remaining dormant until it is triggered to enter the lattice cell. Assembly. During the lactic cycle, newly synthesized phage components, including viral DNA, caps proteins, and tail proteins, are assembled into complete phage particles. Maturation. Assembled phage particles mature and any enzymes required for the infection processed by synthesis. Lysis. In the lactic cycle, once the maturation is complete, the bacteriophage produce enzyme called lysins, which break down the bacterial cell wall. This leads to the lysis or bursting of the bacterial cell, releasing a large number of new phage particles. Release. The newly formed bacteriophages are released, into the environmental where they can infect other bacterial cells and start the cell anew. The lattice cell and cycle results in the destruction of the host bacterial cell. Lysogeny. Lysogenic cycle only. In the lysogenic cycle, in the condition are unfavorable for the lattice cell. Later cycle, the prophage remains integrated in the bacterial chromosome, allowing the bacterium to replicate with the viral DNA as part of its own genome. The prophage can exist the lysogenetic cycle and enter the lattice cycle in certain triggers such as ultraviolet radiations or stress. Induce the process known as induction. The choice between the lactic and lysogenetic cycles is typically determined by environmental conditions and the specific phage involved. Some bacteriophages only follow the lactic cycle, while others may alternate between the two depending on circumstances. The lactic cycle results in the immediate destruction of the host bacterium, while the lysogenetic cycle allows the phage to exist with the host until conditions favor the lactic cycle. Classification of viruses and some viral disease. Viruses can be classified in the various ways based on different criteria such as their genetic material structure, replication cycle, and host range. Here's general classification of viruses based on their genetic material along with some examples of viral diseases caused by each type. DNA causes double-stranded DNA viruses. Example herpes viruses, herpes simplex virus, varicose zoster viruses, adenoviruses, papilloma viruses cause warts and cervical cancers. Single-stranded DNA viruses. Example pyroviruses cause viruses disease, including fifth disease in humans. RNA viruses, double-stranded RNA viruses. Example retroviruses, common cause of gastronutrients in infects the young children. Positive signs single-stranded RNA viruses. Example, coronaviruses. Example, SARS CoV-2, COVID. Picorno example, polio versus hepatitis A versus versus example, Zika virus and dengue virus. Negative signs, single-stranded RNA versus example, influenza versus cause seasonal flu, rabies versus Ebola virus. Retroviruses DNA examples human immunodeficiency virus HIV causes AIDS. Uh, retroviruses have an RNA genome that is reverse transcript into DNA using the enzyme reverse transcriptase. These integrate this DNA into the host genome. Example HIV is the most well-known retrovirus causing acute immunodeficiency syndrome AIDS. Other viruses classification criteria enveloped versus non-enveloped viruses. Some viruses have an outer lipid envelope derived from the host cell membrane, while others lack this envelope. His example influenza viruses is enveloped, while adenoviruses is non-enveloped. Host range. Some viruses are highly specific to certain host specific, while others can infect a broad range of hosts. Example, influenza A virus can infect humans, birds, and other animals, viral families, and genera. Viruses can often grouped into the family and genera based on similarities in genetic material structure and replication cycle. Example, the family flavored includes the genus flavored virus, which contains viruses like Zika virus, and dengue virus. Viral diseases: herpes complex virus (HSV) causes oral and genital herpes. Influenza virus causes seasonal flu. Human papilloma virus (HPV) causes viruses types of warts and is a leading cause of cervical cancer. HIV, human immunodeficiency virus, causes AIDS, acquired immunodeficiency syndrome. Hepatitis B and C viruses cause hepatitis and can lead to liver disease. Measles virus causes measles, a high contagious respiratory disease. Zika virus associated with birth defects and neurological issues. SCARS CoV2 caused COVID 19, a global pandemic. Ebola virus disease EVD, a severe and often fatal illness. Rabies virus cause rabies, a deadly viral incompetence transmitted through animal bites. It's important to note that this is not an exhaustive list of viruses and viral disease, as there are thousands of known viruses, each with its own unique characteristics and are associated disease. Viral classification and disease understanding continues to evolve as new research and discovery are enveloped. Thank you for joining us on this journey through the fascinating life cycle of bacteriophages. We appreciate your support for FahadYounas channel. Please consider sharing, liking, and subscribing to our channel. Thank you for watching my video.